Well, if we concentrate on the phosphoglycerate mutase and analyze enzymes, these are the remaining reactions of glycolysis. They aim to convert the phosphacylester of 3-phosphoglycerate to a high energy form, harvesting the phosphate as ATP. And the 3-phosphoglycerate is first converted to 2-phosphoglycerate by phosphoglycerate mutase, and the 2-phosphoglycerate conversion to phosphoenol pyruvate is catalyzed by the enolase. If we just think about the pyruvate kinase, this is the final reaction of the anaerobic glycolysis and catalyzed by the higher regulated enzyme pyruvate kinase. In this strongly exergonic reaction, the high energy phosphate of phosphoenol pyruvate is just conserved as ATP. And the loss of phosphate by phosphoenol pyruvate leads to the production of pyruvate in an unstable enol form, which spontaneously tautomerizes the more stable keto form of pyruvate and contributes a large proportion of the free energy of the hydrolysis of the phosphoenol pyruvate. We have finished the aerobic glycolytic reactions, but how about anaerobic glycolysis? So the last reaction, especially the pyruvate kinase to pyruvate reaction, is very important in the aerobic glycolysis also, other than the third and first reactions. And what happens in the anaerobic glycolysis is listed in that slide. Under aerobic conditions, the pyruvate in most cells are further metabolized via the Krebs cycle. And under aerobic conditions and in erythrocytes under aerobic conditions, the pyruvate is converted to lactate by the enzyme lactate dehydrogenase, and the lactate is transported out of the cell into the circulation. The conversion of the pyruvate to lactate is under anaerobic conditions provides the cell with a mechanism for the oxidation of nicotinamide dinucleotide reduced form, which is produced during the glass halide 3-phosphate dehydrogenase reaction to nicotinamide dinucleotide, which occurs during the lactate dehydrogenase catalyzed reaction. This reduction is required since nicotinamide dinucleotide is a necessary substrate for glass halide phosphate dehydrogenase, without which glycolysis will stop. Normally, during the aerobic glycolysis, the electrons of cytoplasmic nicotinamide dinucleotide reduced form is transferred to mitochondrial carriers of the oxidative phosphorylation pathway, and it can generate a continuous pool of the cytoplasmic nicotinamide dinucleotide oxidized form. So in aerobic glycolysis, we generate more ATP per mole of glucose oxidized than does the anaerobic glycolysis. So the efficiency or utility of anaerobic glycolysis to a muscle cell means that when it needs large amounts of energy stems from the fact that the rate of the ATP produced from the glycolysis is approximately 100 times faster than from the oxidative phosphorylation. And during the exertion, muscle cells do not need to energize anaerobic reaction pathways, so the requirement to generate the maximum energy amount of ATP for muscle contraction is in the shortest time frame, and this is why muscle cells derive almost all of the ATP consumed during the exertion from the anaerobic glycolysis. Yes, the lecture is finished.